Hello everyone, my name is Gina Wang. My project is on LUX-I-LUX R-type quorum sensor system in K61, the current species novel growth and AHR production in magnesium sulfate-rich brine. And now let me explain those big words in the title. Before I go into the bacteria I was studying, I would like to first introduce the plant here, taro. Taro or kalo in Hawaiian has an important historical and cultural impact in Hawaii. And now here's the reason why I talk about taro first. The bacteria my research focuses on, K61, a novel Dicera species that is expected to be named Dicera wulopoensis, was isolated from Eloi at the Wulopo heiau on the eastern edge of Kauai Nui Marsh in Kailua, Hawaii. And this is one of the places where you can find an abundance of taros. The scientists I am working with are also investigating the potential of planting taros on Mars. But before bringing anything on Mars, we must learn as much about them and as many reactions and interactions of them as possible. The potential of K61 surviving on Mars and its survival mechanisms, such as quorum sensing, are also important to understand. In this research, we tested K61's viability and reaction in magnesium sulfate-rich brine. You might be wondering why we chose magnesium sulfate-rich brine. It was chosen because it has both astrobiological and agricultural implications. As I mentioned in the previous slide, taros have the potential of being planted on Mars, and since magnesium sulfate-rich brine has been present on Mars, both in its history and in today, bacteria's tolerance level to this brine is significant for forward contamination, which is the transport of Earth-based microbes to other planets. In an agricultural context, magnesium sulfate may play an important role as plant fertilizer by supplementing magnesium to deficient soil. Some relatives of K61 are plant pathogens, such as Dicera dodonti. However, there is no evidence yet on whether K61 is pathogenic or symbiotic to taros. Nevertheless, finding out whether magnesium sulfate promotes or inhibits K61 has implications for taro both on Earth and on Mars. We also wanted to study quorum sensing inside K61. Quorum sensing is a way for bacteria to communicate. LUX-I, LUX-R type quorum sensing is one type of quorum sensing, which this study focuses on, and here's how it works. LUX-I is a gene. When it is transcribed into protein, it acts like the mouth of the bacteria and sends out a chemical signal called acylhormoserine lactone, or AHL for short. These signals can then move freely between cell membranes. LUX-R is also a gene. When it is transcribed into protein, it acts like the ear of the bacteria or receptors of AHLs. Once the signal, AHL, accumulates to a certain threshold, the receptors will respond by activating the transcription of target genes. The concentration of AHLs reflect the number of cells. Because the more cells there are, the more mouth LUX-I there will be, and the more AHL signals will be sent. So if there is a low cell density, there are few AHLs, and the gene transcription will not be activated. However, at high cell density, there are sufficient amounts of AHLs, so the gene transcription will be activated. This way of communication is essential for bacterial activities, such as biofilm. A biofilm consists of many cells stuck together onto a surface. These microbial cells are enclosed within a matrix composed of polysaccharidoses, proteins, and so on. Biofilm helps with bacterial survival, especially against harsh environments, such as high salinities. It also promotes bacterial growth because it provides them an optimal environment to replicate. It is important to learn whether K61 utilizes quorum sensing and how this process works so that we might know how to promote or inhibit the growth of K61 in response to future interaction between K61 and taro. So here are my research questions. Question 1 was, in what percentages of magnesium sulfate-rich brine could K61 survive in? Question 2 was, does K61 produce biofilm in normal conditions or in magnesium sulfate-rich brine? Question 3 was, does K61 utilize LUX-I, LUX-R type quorum sensing? Question 4 was, what types of AHLs does K61 produce, and would this change in magnesium sulfate-rich brine? To find out what percentages of magnesium sulfate-rich brine could K61 survive in, we grew K61 in various concentrations of magnesium sulfate-rich brine. K61's growth was determined by measuring the change in optical density by spectrophotometer, because the more bacteria there are, the more dense the solution will be, and the more light will be blocked. Among the concentrations tested, 0 to 5% magnesium sulfate-rich brine had approximately the same optical density after 24 hours. The optical density of 10% magnesium sulfate was lower but still enough to show growth. It should be noted that growth was observed in 15% after 1 week, in 20% after 2 weeks, and in 25% after approximately 3 weeks. As the pictures show, in 10% magnesium sulfate-rich brine, a pink thin layer of biofilm was formed at the air-liquid interface. In 15%, the pink biofilm layer is extremely thick. But in 0%, biofilm was not observed. So the answer to my second research question was that K61 produces biofilm in magnesium sulfate-rich brine, but not in normal condition. To answer our third research question, does K61 utilize LUX-I type quorum sensing? K61 and the biosensor CV026 will streak perpendicular on agar plates. CV026 is a biosensor that only produces violacin, which is a purple pigment, when certain types of AHLs are being detected. Based on these purple pigments on an agar plate, we can conclude that K61 does produce AHLs and utilize LUX-I type quorum sensing. Another way to determine whether K61 utilizes LUX-I type quorum sensing is through bioinformatics tools. K61's genome was annotated so that 1 LUX-I gene and 7 LUX-R genes were identified. So our next step is to find out what types of AHLs K61 produce. We extracted AHLs produced by K61 and used liquid chromatography mass spectrometry, or LCMS, to determine their types and relative amounts. Because LCMS is a targeted analytical technique, 10 AHL standards in this table were used. Only AHLs in this table could be detected in our experiment. Among these standards, 5 types of AHLs were detected, including C6, C6-oxo, C7, C10, and C12, which are highlighted in this table. We then wanted to test whether the types of AHLs produced by K61 would change in magnesium sulfate-rich brine. Given the time constraint of this experiment, we chose to work with a 10% magnesium sulfate-rich brine solution. So in triplicates, K61 was cultured in the control group, which was no or 0% magnesium sulfate-rich brine, and the experimental group, which was 10%. The optical densities were measured at various stages in K61's life cycle to create growth curves. At 24, 48, and 72 hours, ethyl acetate was added so that 2 layers were formed. The top near transparent layer is ethyl acetate with AHL dissolved inside it, and the orange bottom layer is cell debris. The cell debris layer would then drain out. The remaining solutions were centrifuged in smaller vacuum tubes for further sedimentation, and the cell debris layers were once again drained out. The remaining solutions were then poured into round bottom flasks. Then by using a rotary evaporator, we were able to get rid of most of the ethyl acetate, so now it's just the lipids remaining, and AHLs are lipids. The types and relative amounts of AHLs were then identified from the remaining lipids by LCMS. So the graph over here has the growth curves of the experimental group, which is in red, and the control group, which is in blue. The x-axis is the time in hour. The y-axis is the absorbance of K61 at 525 nanometer. The two groups reach its exponential phase at approximately the same time. However, for the experimental group with 10% magnesium sulfate-rich brine, most cells were present at the mid-late exponential phase, and at around 72 hours, the control group reached its carrying capacity, but K61 in the experimental group continued growing. K61 appears to like 10% magnesium sulfate-rich brine. And here are the results of AHL extraction. The x-axis is the time in hour, and the y-axis is the absolute peak area of AHL. In both groups, K61 produced C6-oxo and C6. These two AHL show an inverse relationship during K61's growth. C6-oxo is produced less as K61 nears carrying capacity, while C6 is produced more as K61 nears carrying capacity. And in the experimental group, C7, C10, and C12 were no longer produced, but C8, C14-oxo, and C14 were produced instead. So currently, Dr. Dobell has done a pathogenicity test on K61 and potato, which is also a tuber plant like taro. K61 was injected into the stem of potatoes through a coated wound, and none of the potatoes show any symptoms. Therefore, K61 is likely to not be detrimental to taros. For future research, we can continue to investigate K61's pathogenicity, such as whether it would change under different temperatures or other environmental conditions, and whether K61 is beneficial to taros or not. We could also monitor the growth of K61 in higher concentrations of magnesium sulfate-rich brine, and investigate the transcriptomes of K61 in different phases and different environments, and perhaps test out how K61 responds to other environmental conditions found on Mars. K61's abundance growth in 10% indicates that it is absolute tolerant and might even benefit from the presence of magnesium sulfate. This has great significance for the forward contamination of Mars and fertilizer use of magnesium sulfate, since C8, C14-oxo, and C14 were produced in the experimental but not in the control group. These AHLs could be important to K61 survival in high salinities. There's so many unknowns and potential out there in space. Studying new and exciting bacteria like K61 will help us explore the potential of life in space. It will help lay the foundation for humanity to plant taros or other greens in space. Through my experience, I learned how important it is to communicate with your mentors and ask questions when needed. My mentor helped me greatly and supported me through the entire process, and I really learned a lot from them. I also learned that you should try to enjoy the process of doing research and then presenting results at science fairs. Through science fairs, I have learned about projects that are very interesting, creative, and in a really wide range of subjects. I also met peers and judges who love science like me and share my own project and results with them. The more you enjoy this process, the more you will learn. I would like to give special thanks to Mr. Jack K, the K Fellowship Program, and Iolani School for sponsoring this research, and the University of Hawaii at Manoa and Donaghy Lab for providing equipment and a lab for me to work in. I would like to extend my great gratitude to Ms. Christy Handel and Dr. Rebecca Prescott for mentoring my research and experiment in the lab, to Dr. Yvonne Chan for guiding me through the complicated paperwork and having weekly meetings with me to ensure smooth progress, and to Dr. Stuart Donaghy for opening his lab's capabilities to me. I'm also grateful to Mr. Timothy O'Donnell for performing chemical analysis, to Dr. Jennifer Saito and Mr. Sean Larry for helping me and supporting my lab work, to Mr. Steve Case and Ms. Lemomi Kikina for helping me to pass the biosafety training and get my certificate, and to Dr. Shafari Dubal for data regarding K61's pathogenicity. This research would not have been able to happen without the support and help from these individuals. Thank you so much. Here are my citations, and thank you.